So we're continuing to talk about tests of selection using polymorphic marker data. The key features are that we're not making any assumptions about traits. These are trait independent, and we are basically taking a sample from a population. What I want to talk about is to conclude these tests by talking about areas where there's a large number, those based upon the site frequency spectrum, and those tests based upon linkage disequilibrium. But as always, any questions about anything at all we've discussed so far? Yes?
Can you explain false discovery rate again?
Sure. So false discovery rate is simply the notion that a discovery is a test I declare to be significant. So I use some criteria, and I can adjust that criteria to put more or fewer elements in that set. So imagine here, all you are markers, and this first row are all markers that by some criteria I declare to be significant. The false discovery rate on this set would be, among this test, what fraction do I expect to be false? That is, to not be deviations from the null, that is just random things that by chance I put in the significant set. So false discovery says, if the false discovery rate, let's say, is 5%, and I have 100 markers that I declare to be significant, it means 95 of those markers, on average, are expected to show true significant departures from the null distribution, and 5% are ones from the null, that is no selective effects, that simply by chance get included. And the powerful thing about false discovery is I can change the discovery set size. And what the false discovery rate does is it changes that size until it makes that set large enough to have a certain number of false discoveries. And the importance is that if I use something like Bonferroni corrections, which are terrible, in a Bonferroni correction, for example, there's an example I give in the appendix, we're looking at breast cancer genes. And I think in the Bonferroni setting, they, with expression data, detect about 55 to be significant with Bonferroni. If you use false discovery, at the same control rate, you detect 140. So false discovery rates are far less stringent because you're not worried about, oh, don't put a bad result in, don't put a bad result in, don't put a bad result in. What you're worried about is, given a certain set, how large can that set of discoveries be so that the fraction of the things I declare to be discoveries that are not true discoveries or false discoveries is at a preset level. And that's a shuttle. It sounds really similar and it's really subtle, but that shift in thinking is actually quite powerful. And again, the motivation for it is Bonferroni basically assumes life sucks and basically almost everything you look at is from the null distribution. So you're really worried about throwing stuff that's not in there. False discovery says a certain reasonable fraction are not from the null distribution, and therefore you want to pick a set that has a high probability of including those with the smallest amount of error possible. So for most genomic studies now, people go to false discovery rates. Yeah? 5%. There's no magic number. All 5% says is 5% of these discoveries are false. You could set it at 1%, you could set it at 40%, right? You can set it whatever you want. Just like a p-value doesn't make any sense. P-value, what's so magical about 5%? Nothing, right? So that's why people typically report p-values instead of significant values. You can also do what are called q-values, and a q-value says if you have this specific test statistic, you can then, a q-value is what's the chance it's a false discovery. So it's a bit different. False discovery says in this set, I use some criteria, in this set the false discovery rate is 5%. I can go to a specific test value and say your false discovery rate is 2%. Or more correctly, tests that are your value or more extreme have only a 2% chance of false discovery. And that's a concept called Story's q-value. So there's a p-value, and a q-value basically is what's the probability the test of this size is a false discovery. The nice thing about false discovery is it's very specific to your particular set of tests. If you go to a different set of tests or observations, false discovery rates are all different. So the nice thing is it's very particular to your particular set. And again, gory details in Appendix 4.
Is false discovery universally better to use? Or is this very confidential?
Well, the answer is it depends. And it really basically comes down to this. is your intuition that almost everything you're testing is going to be from the null. And the null, by the way, could be in a drug setting, not that there's no difference, but that it's no difference or worse. So if your concern is that almost all of the things you're testing are going to be false, and you're really paranoid about declaring one of them to be good, because you declare. So we can have different estimators of theta. Remember, theta is the thing that completely describes this process under the equilibrium neutral model. We've seen before, I can take the number of segregating sites S, and if I divide that by this thing here, which is entirely a function of the population, the sample size, I can get an estimate of theta. That's often called the Watterson estimator. I can take pi, the nucleotide diversity, the average number of pairwise differences. That's often called Tajima's estimator. And then I've got these two estimators here. Zeta, so zeta is basically the unfolded spectrum. This is the number of singletons. So if I take the fraction of sites that have only one copy of the derived allele, that's an estimate of theta. If I take the fraction of sites where they have only one copy of the minor allele, that's a slight correction, but that also gives me estimates. So I can estimate it from segregating sites, diversity, or singletons. Either these don't require unfolded data, these two do. For large sample sizes, they're essentially the same, but basically, this is for unfolded, this is for folded. Unfolded means I can tell you which allele at that SNP arose by mutation, as I can polarize it. Folded means I don't know, I simply take the minor allele. So this is the number of times sites that have only one copy of the minor allele. This is the number of sites that have only one copy of the derived allele. Those are called singleton estimates. So here are kind of the standard site frequency tests. And as you can see, and I will tell you in a second how you can make your own site frequency tests. You can have, Jill's site frequency tests or Bob's site frequency tests. So Tajima's D test, so you see that it requires folded or unfolded. You see a lot of the tests require simply folded. This is comparing the estimator based upon segregating sites with the estimator based upon nucleotide diversity. A value less than 0 means I have an excess of rare alleles. That's either a sweep or a population bottleneck. A value bigger than 0, I have an excess of intermediate frequency alleles. That means either balancing selection or population structure. Someone asked about sequencing errors. You've got Y and Y star, which is basically Tajima's test here, except what I'm contrasting is the number of segregating sites where I've removed the number of sites which have minor allele singletons. So this is the folded spectrum. I take S here and I throw out all the things that exist as one copy as a minor allele, and I then compute pi the same way. So if you're worried that you're getting sequencing errors would typically appear as a singleton. That is one error, give you a sequence you don't necessarily see. So this adjusts for that, and then Y is that same adjustment, except now I'm using, instead of sites with one minor allele, I'm using sites with only one derived allele. So this is the first unfolded. Then we've got other comparisons. So Tajima's is segregating sites with respect to polymorphism. So Fu and Lee's D and D star. So the star means folded. So this is comparing segregating sites with the number of derived singletons, segregating sites with the number of singletons based upon minor allele frequency. They're F and F star, again, the comparison. So we have three things to compare, S, pi, and theta 1. Theta 1 can be minor alleles, so it's folded or alleles with one, derived alleles with one copy, so it's unfolded. So here the comparison is, again, now instead of S with pi with singletons, pi with singletons. So basically these are different ways to look at this comparison. So these are all pretty similar. They all have the same features as Tajima's. Two relatively recent ones, which use much more accurately use the power of the unfolded spectrum. Fei and Wu's H contrasting the estimate of theta based upon nucleotide diversity with this estimate here, which basically weighs derived alleles at higher frequencies. More will come back and talk about that. And the same thing for E as an estimate. We'll show you where all these things come from. It's actually pretty straightforward. So in Fay and Wu's H, a value less than 0 means you have an excess of high-frequency derived alleles. That's either a sweep or allelic surfing. It's not caused by a bottleneck. It's a feature largely independent of demography. Here, a value less than 0 means I've got an excess of low versus high-frequency derived alleles, and this is a signature of a past sweep, a sweep that has occurred. There's a short window where that little lingering shadow of the sweep appears on this criteria. Remember, all these methods are specific to a certain time window. Some catch ongoing sweeps, some catch just completed sweeps, some catch the slight shadow of a sweep that's been around for a little bit. So the tests all have kind of different windows that they look in. So yeah, question. Yes, I'll talk about it. Basically, we'll show a little bit later on. It's on the order of 0.1 NE to 0.4 NEs. So it's set by the population structure. Yeah, this is the overview. We'll talk about, kind of drill down on more on this. And again, any details I don't mention in here, you can certainly clarify them, or they're all spelled out in detail in the book. So the basic structure of the tests are, I have two different estimators. I compare the estimators, and I then divide by a standard error for those estimators. So now let's see how we can put all those tests together. So here is the typical data that I have. So if I'm looking at the derived alleles, the expected number of sites that have I derived alleles is theta over I. The expected number of sites that have I copies of the minor allele, well, I could have I derived or N minus I derived. I get these expressions here. So if you think about that, if I have derived alleles, I just rearrange this, and an estimate of theta is simply I times the number of sites that have I derived alleles. An estimate of theta, if I look at minor alleles, is I, number of sites with I minor alleles, times N minus I. So this is my observations, number of sites with I minus alleles, then I correct for I times N minus I over N. So each individual class, singletons, 2 copies, 3 copies, 4 copies, each individual class can be used to give you an estimate of theta. Suppose I want to estimate theta based upon sites that have 5 derived alleles. Well, the estimate of theta is 5 times the fraction of sites with 5 derived alleles. My estimate of theta for sites with 5 minor alleles is 5 times N minus 5 divided by N times the number of sites with 5 minus alleles. So basically, each category in the site frequency spectrum, either folded or unfolded, can be used to estimate theta. And the various tests I've talked about just weigh these categories differently. That's all they do. So here is the basic approach. I have some, we'll just call this some estimator G. It's some weight times the number of counts. I then, if I put in what the expected value this is, it's simply theta over I. I pulled the theta out in front. So basically my estimate of theta for any weight scheme I want is my estimator divided by this thing here, which simply gives you a balance on the weights. Why? Because this theta comes out front. So, for example, if I take C, if I weight every site equally, that's just counting up the number of segregating sites. That gives me S. If I then plug this in, 1 over I, that's that AI expression I had before. If I take C as these weights, that then gives me my Tajima estimator. If I take the weights as looking at C1 equals 1, everything is 0, that's the number of derived singletons, and I can do the exact same thing now with the folded frequency spectrum. So all these tests you see up here are basically taking this site frequency spectrum and weighting the points. So that leads us down to Fay and Wu and Zhang's H and E statistics. So we'll come back to that in half a second. So, questions so far? Again, kind of giving you an overview. The critical part of the overview isn't the math about how you make the corrections. The critical part is that each individual class by itself, the number in that class, can be used to give you an estimate of theta. And different approaches basically amalgamate that information differently. So we're looking at different comparisons. So the classic one is Tajima, and Tajima's d-test is basically this. And that expression down there, where S is the number of segregating sites, is basically figuring out what the variance on these things are. So a negative value of D means there are too many low-frequency sites. Positive value, too many intermediate frequency. Expressed another way, D is a test for whether the amount of heterozygosity per site, there we are, is consistent with the number of polymorphic sites under the equilibrium neutral model. Under selection sweep or population expansion, heterozygosity should be significantly less than the predicted number of polymorphisms. Why? Under a sweep or expansion, you have a lot of singletons. So Fay and Wu's H-test, the weights that are now given are I squared. So this is now for the unfolded frequency spectrum. So I can weight high values. What this simply says is as you get more and more, have a site with a higher and higher fraction of derived alleles, you weight it more. And the idea is one of the things that happens under selection is you get a shift, you get a lot of high frequency derived alleles. That's a very unusual signal you don't get from most demographic features. You can get it from a surf, but not from anything else. So this is one weighting scheme they had to use that, and then basically that's their estimate where you're based upon theta by overweighting the high frequency values. And what they basically showed with simulations, here's the power of the H-test and the power of Tajima's D, and this gets at your question over here. And here is where that signal persists. So basically, after about 0.1 generations, you don't have a lot of power from the H-test. As the sweep is just concluded, the H-test has a lot of power. One reason is those high-frequency alleles get fixed very quickly because they're near 1. The Tajima's D-test over here actually has a lot more power as you go a little bit further. So the way to apply these tests is you can use Tajima's D and either the D or the E test. So the E test here, instead of weighting high frequencies by I squared, weights them by I. No reason for one or the other. People try different weightings and see which had a better signal. So what we basically then do here is you compare this weighting of high frequency against number of segregating sites, and H here compares the high frequency with respect to diversity. So these, again, different contrasts within that, and the bottom line is that the E test is regarded as, the E and H tests are regarded as pretty good tests for detecting if sweeps have occurred, and they're pretty good because they're robust to many of the issues that come up with demography. And the reason is it's easy to get a lot of low-frequency minor alleles under a population structure. It's very hard to get a lot of high-frequency derived alleles under anything other than a sweep. So that's why you have these various tests. So there's the family of tests. Like I said, you can make your own test. You can go in and figure out which weights you want to put on those, and that's your own magical test. So questions about these site frequency spectrum tests. Basically, looking at different parts of the site frequency and asking if they're consistent. Well, so the problems you have here is unfolded has more power because high frequency derived sites are a feature you don't expect under demography. The problem is you have to polarize the data. You have to have an outgroup. Not necessarily a big deal, but with unfolded data, you don't have to worry about that whatsoever. Unfolded data, your polymorphic sample is sufficient to use for folded data. For unfolded data, you also have to sequence an outgroup. Not necessarily a big deal, but you know, it's... Yeah. Oh, thank you. Sure. Yeah, so evolve, people know about evolve and resequence data, resequence tests. So basically, there's several ways you can do it. You basically take a population, you sequence it and get allele frequencies, then you allow it to undergo selection. Either on its own, you throw it in the harsh environment, let it go, or you actually target specific traits with standard artificial selection. Then you go back and you resequence it and see what's changed. You can certainly use these approaches. In evolve and resequence, I think you're better off looking at for allele frequency changes because you've got those values beforehand. You can act, and since you know something about the affected population size, you can come back and get those. Now, what you can do is you can use Tajima's test for a quick and dirty scan, but I think you're better off looking at allele frequency changes. Yeah, sure. SNP filtering bad for these tests. So if you use a SNP chip, you get biased results because SNP chips are designed to filter out low-frequency SNPs. Low-frequency SNPs give you a lot of signal in these things. So the data you require for these are different from, let's say, a data for a GWAS, because you need those low-frequency SNPs, because that's where a lot of power is. Or conversely, a high-frequency derived allele is still regarded as a low-frequency SNP because the SNPs aren't polarized. So that's the thing, is they are important. So singletons, as we'll see, have a lot of signal in them. So as sequencing gets cheaper, so your error rates go down either because you're doing shorter, more accurate reads, or you're doing, gazillion level coverage. Then you can start using the singletons. And the singletons give you a lot of information. Or singletons, I mean rare SNPs. And when you talk about a rare SNP, we simply mean a SNP where the minor allele frequency is rare. Some of those are high-frequency derived SNPs. You don't want to throw those out because those have a lot of signal. No, you're absolutely right. So again, the technology you're using depends upon the question you're asking. And so there's some discussion in the book about ascertainment bias from SNPs. That's a historical artifact that will go away as people start, doing genotyping by sequencing or whatever and getting, just not using a SNP chip, but just getting sequenced directly. And having appropriate filters to keep all those SNPs in there. Again, if you're worried about sequencing errors, you can always do these, which basically throw out singletons and ask what happens. And so a good way to do it is to use, let's say, Tajima's, and then use one of these two and ask how different they are. If they're rather different, it means those singletons are important. That could be because you have a lot of sequencing error, or it could be because you've got selection. So you can always do internal checks in your data to see that. You should always, you should never run just one test on your data. So we have this issue of these site frequency tests are very dependent upon the equilibrium assumption. How do we get around this equilibrium assumption? Well, there are 4 approaches people use. Number 1, the outlier approach. I compute, just like with FST, I compute a whole bunch of D statistics, I get a distribution, I focus on the outliers, the most negative ones, for example, for directional selection. Number 2 is I could use the marker information to reconstruct the ancestral history, bottlenecks, etc., and then do a coalescent based upon that history, and then ask how unusual my particular observations are given that simulation of the null model. I could use the empirical site frequency spectrum. So I pick a set of markers I think are random, use that as the site frequency spectrum, then compare that to the data I see. The key part about that is if your null is background selection, which is the appropriate null, if I'm comparing, let's say, a target gene in a certain chromosome, I then want to compare it to an empirical site frequency spectrum where those markers are drawn from regions where there's a similar gene density to recombination region. As you change gene density to recombination, you change the strength of background selection. Then you have support via a preponderance of evidence, right? Death by 1,000 cuts. Looks at the joint signature of a bunch of different tests. Let's look quickly at these. Here are D values for African and European Americans. The histograms are given by here. The dashed curve basically is the best fit given a coalescent simulation. And what you'll notice here is that Africans have a negative D value. Europeans have a slightly positive D value. So if you get a negative to GMAS D, in Africa it's harder to interpret because the whole distribution is shifted to negative values. You get a negative D where the distribution is shifted to positive values, it actually gives you a bit more support. Now what you're probably thinking is, well, how come I just can't subtract off the mean and then use it that way? The problem is you haven't adjusted for the variance. So when you have departures from the equilibrium model, you also inflate the variance, and so you expect more outliers. So usually when people do outliers, for example, these are probably really good candidates for genes under selection. Why? Number one, they're outliers. Number two, the whole distribution is shifted to positive values. So these are even more extreme than you might think. Over here, probably not a lot of evidence for there because the distribution is shifted left to begin with, and just because you're an outlier over here, notice too they're not really that much of extreme outliers. So you can look at outliers and try to adjust outliers by that, but it's very ad hoc. You can use coalescent simulations to try to clean that up a little bit, but the problem with the coalescent simulation is you could build in a bottleneck, but if you've got a population expanding in space and don't account for allelic surfing, your coalescent isn't appropriate. I mentioned about using the empirical site frequency spectrum. A couple of tests you might see are MWU low, MW high. This stands for Mann-Whitney U test. It's a non-parametric test. So what Rasmus Nilsson did here basically is they created an empirical site frequency spectrum and then looked if the target site with respect to that empirical spectrum was overrepresented in the high values or overrepresented in the low values. So the idea by using the empirical frequency spectrum is you're not using Watterson, which assumes an equilibrium distribution, but rather you're trying to let the data speak as to what its past history is. So again, all these approaches are logical and reasonable, but they're not necessarily approaches that are statistically rigorous in the sense of giving you good p-values. That's the take-home message for most tests of selection, is they're honest efforts, but in most cases you can't put legitimate p-values on these things. So support via preponderance of evidence, I'll skip over most of these. The idea is that I do multiple tests and I look and see if those multiple tests give concordant signals. So one approach, the CMS, composite of multiple signals approach, basically looked at several different tests and used those to refine location. The problem with these is many tests are correlated, not independent. So if I do 5 different site frequency spectrum tests on the data, I expect those to be correlated. But if I do a site frequency test, an FST test, a cross-population test, those are a bit more independent, and I maybe feel better about the evidence. So what these tests basically do is try to adjust for correlations, but the bottom line is if you just randomly generate coalescence under drift, you're going to see some outliers. And a region that has an outlier coalescent will have a number of tests which appear to be unusual. And you need to distinguish that random event from the neutral coalescent from an actual event. So there's a couple of ways. I'll just talk about this really quickly. So one way is you can basically rank the test statistics and then use a z-score method, compute them to normal random variables. Again, the gory details are in the book. I want to kind of give you a feel. Another way is a great name, deconvoluted composite of multiple signals. This is a cool idea. Let's say I have 100, 1,000, I've done 500 markers, I've done 10 tests. What I can do is treat that as data. And I can look at, over that set of markers, how correlated tests are. So it's simply treated as data. I've got a big data matrix. So a column is a given test. A row is a given marker. I can look for correlations across tests at a given marker. So I can put all those in, and this is kind of a scheme that basically weights the empirical correlation that you observe, and then basically generates a test statistic that accounts for the fact that some tests are more correlated than other in your random sample, and then asks, is your particular thing showing excess of those correlations or not? My favorite one, which is a big mouthful, again coming out of Mike Whitlatt's group, has the great name of Mahalanobis distance based on negative log rank p-values, MD rank p. That's a mouthful. But it simply means this. Again, this expression should look familiar. Transpose a vector, inverse variance covariance matrix, a vector. That's a general distance metric. The idea is, if you look at outliers, you usually think about one dimension. That's in the tail, that's in the tail. If you look in 2 dimensions, the data are in an ellipse typically. And that ellipse is given by the covariance structure. So in 2 dimensions, you've got to weigh outliers differently. And some outliers are more important than others, because if they depart from that ellipse, they're much more of an outlier than at the same distance on that ellipse. Extending that to as many dimensions as you want, the Mahalanobis distance is a way to basically look for how far apart 2 points in multivariate space are, where I adjust for their covariance structure. So this approach here is basically, again, looking at correlations among the tests and returning a test statistic based upon, for a given marker, how far those tests are away from what you expect the observed covariance structure to be. So again, there are details here, but basically all these are approaches that try to take your observed test statistics and their correlations and figure out how to combine them. So, summary of site frequencies spectrum. To G minus D, most often seen, negative values can be generated following a sweep. The problem is change in population size also generates that. The H and the E tests, so Fei-Enwu's H or Zhang's E test, these are high-frequency derived alleles. These are more powerful because they're a signal you don't expect under most tomography, but they're also more fleeting. Because they're high-frequency derived alleles, once the sweep stops, allele at high frequency gets fixed by drift fairly quickly, the signal goes away. So those are site frequency-based tests. They have a long history, they're widely used. Nowadays, they're probably more used as a summary statistic of the data, like you almost always report it to G minus D when you're looking at a marker, because it gives you some information. Is there some structure? That structure could be from selection, or it could be from a population bottleneck. A D over a whole bunch of markers tells you something. So for example, what can we say in here? In Africa, the negative D here suggests a population expansion. So that's because the entire set are all shifted one direction, and that's consistent with coming out of a bottleneck. So questions about site frequency tests. There's a bunch of them, but as you see, they're all connected to using different bits of information weighted differently from parts of that site frequency spectrum, either folded based on minor allele frequency or unfolded based on frequency of derived alleles, which you polarize within our group. So the last category of tests are haplotype-based tests. And these are probably among the most powerful tests for detecting ongoing selection. They've got features that make them more robust to issues of demography, and these are the ones you mainly see, for example, in the human literature. So I'll talk about some old school approaches, allele frequency spectrum based upon the sampling formula. I'll talk about linkage disequilibrium, giving you different signals for soft versus hard sweeps, and then a bunch of tests basically asking, are there features of the alleles which are inconsistent with their frequency? Under neutrality, a common allele is an old allele. An old allele should have a smaller haplotype associated with it. There's a bunch of tests in here. These are among the most powerful for detecting ongoing selection. We first of all have to figure out what a haplotype is. What I do, basically, is I take a core area, either one SNP or a couple of closely related SNPs, and then I define that combination as my allele. Then I take that core haplotype and I move out from that, and I look at how the sequence varies as I move out. So how long do I go between two sequences that share the same SNP haplotype before the sequences differ by one site or differ at 5%? So there are lots of metrics that basically look out, extending out from that core haplotype and asking how sequence variation changes. So the basic overview is, if I have a certain number of segregating sites, do I have too many or too few haplotypes? You could imagine one extreme where I've got a whole bunch of segregating sites, but only two haplotypes. They just differ in those segregating sites. Or you could imagine that the number of haplotypes equals the number of segregating sites, right? So those are both examples of extreme structure. Strong haplotype structure occurs when there are fewer haplotypes expected given the number of segregating sites. So under dispersion of haplotypes, that's a signal of excess LD. It results in a deficiency of haplotype diversity, which is kind of the equivalent of pi, infinite sites model. Such a signal can be generated by a partial sweep, recovery from a bottleneck, balancing selection, or population structure. I could also have the opposite pattern, an over-dispersion of haplotypes, too many haplotypes relative to the number of segregating sites, and that results basically from a hard sweep or recovery from an extreme bottleneck. So just kind of an overview of those complete or nearly completed sweeps. I have excess number of haplotypes, excess diversity, excess of high-frequency haplotypes, LD structure, high LD on either side of the site, little across it, a partial sweep or a recent balancing selection, strong haplotype structure, deficiency of number of haplotypes, deficiency of diversity, excess of low-frequency haplotypes. LD structure is long haplotypes at excessive frequencies, allelic age, long haplotypes, excessive frequency. A soft sweep. Remember, a soft sweep is I either have a site that's been existing for quite a while before it's under selection, so I've got multiple haplotypes with that favored site that as a group get increased in frequency. So I've got moderate haplotype structure with a few dominant haplotypes, not just one, but they're not equally spread out as well. Then the LD structure is high pairwise across the entire regions. So these are all signals we're going to be using, right? And what I've done in the chapters are to summarize the overall features before going into some of the tests. We've seen UN sampling formula before. If I know theta, I can get a probability distribution for the number of alleles given the sample size in theta. I can basically ask, are there too many or too few alleles given my estimate of theta? I can go a step further in that if I condition on the number of alleles I see, theta drops out, and the allele frequency spectrum basically is simply a function of the number of alleles I see and the sample size. And then I can use that for various tests. So the initial test that people looked at was they computed this test statistic given their data, and they compared that value under what I expect from the UN sampling formula, asking if it was extreme or not extreme. This is a Shannon-Weaver diversity index, but Watterson, this is what UN suggested. Watterson suggests this test based basically upon, those are basically frequency of homozygous was a bit stronger. This is called the UN's Watterson test or homozygosity test. This is one of the early tests that people use, one of the first molecular tests based mainly upon protein data. The idea is you compute this test statistic based upon your data, then you go back and use the Watterson distribution and ask what the value of this test statistic should be under the Watterson distribution, and then ask if they significantly depart from each other. So it's an equilibrium test, because when I go back and use the UN sampling formula, use the UN sampling formula, that's an equilibrium formula. Just like Watterson is the equilibrium value for the site frequency spectrum, UN's is the equilibrium value for the allele frequency spectrum. These are kind of old school tests that, aren't used much more, but they're historically important. So these tests here, I simply use the site frequency spectrum conditioning on K, so I don't need to know theta. The next group of tests is I can test for whether there's too many or too few alleles, and I do that by having an estimate of theta, and for theta, what I typically use is I use the Waterson estimator, theta based on the number of segregating sites. I can basically ask, is that too many or too few? And that's the foo W test for too few, and the foo S test for too many, which is basically just the fact. So there are tests I can use based upon the the allele frequency spectrum, whether there's too many or too few alleles. The category of tests which seem to be fairly nice are these haplotype diversity tests, because they've now been extended to soft sweeps. So here, this is just, what is this value here? This is simply the expected homozygosity. This is a measure of diversity. So if I pick two sequences at random, what's the chance that they differ? This is the chance the two sequences at random are the same. So 1 minus that's the chance they differ. So that's often called the haplotype diversity, or H test. We can then contrast that value with the value we expect under a simulation. But the importance of that is it's been extended, and the H1, 2, and the H2 tests are recent extensions, 2014, to deal with soft sweeps. And the idea here is H1 is simply the test I had here, but H2 says take the two most common haplotypes. There they are. So, and lump them together. So those are the two most common, and there's everything else. And the idea was that if I have a soft sweep and it's not too soft, I may not get one dominant haplotype, but I'll get a couple of dominant haplotypes. So I put those in, and so the ratio of H12 to H1 is often used that. So for a hard sweep, H12 is about equal to H1. For a soft sweep, this is a statistic here based upon counting the second and third and fourth most frequent. The H2 to H1 ratio gives me some idea. So for a hard or soft sweep, this ratio should be bigger than 1. Right? A soft sweep has two haplotypes that get lumped together. A hard sweep has one. So again, when I go through a lot of tests like this, it's a lot of information. So what should you take away? What you should take away is the basic rough idea, because you can go back and look these up. And the rough idea is by counting the frequency of haplotypes in your sample, I can look at the frequency of homozygotes, and therefore I can compute what's the chance that a random draw is going to give me two different haplotypes. That's what the H1 test does. The H2 test says take the two most common haplotypes and lump them as one, and then redo the same procedure. And ratios of H1 to H2 and the H12 to H2 are tests that give you signals whether or not you have a hard sweep or a soft sweep. So basically, there's a bunch of stuff here. Here are some of the tests based upon the allele frequency spectrum. The Waterson test, I mentioned about that. We have an exact test which basically compares the observed with the expected full distribution. We've got tests for too many haplotypes. We've got tests for a deficiency or an excess of rare haplotypes. We have the observed number of haplotypes. We've got the observed haplotype diversity given S. We've got these H12 and H2 tests. There's a bunch of these, and I want to give you the basic field the book talks about in detail. So we're not really concerned with that. The idea is I have a sample. Here's my sample, and I'm simply asking if I take that sample and I redraw from that sample, how often do I get the same sequence? Not in the population what happens. And the idea is that if I have strong haplotype structure, I'm going to have a dominant haplotype and a bunch of noise. So if you look at that haplotype homozygosity will be really big. You think about what happens if all haplotypes are equally frequent, then pi is 1 over n. So 1 over n squared is basically 1 minus 1 over n. You don't get much of a signal. If you've got strong haplotypes, so almost all the haplotypes are 1, then that signal is very close to 0, that h. So you basically, that's kind of the signal you're looking at. The idea is we're trying to look at different ways in single numbers to quantify strong versus weak haplotype structure. So those are all tests based upon the allele frequency spectrum. And the book talks about those in detail and gives you the original references and some of the concerns with that. Now we can have tests based upon linkage disequilibrium. And remember, here's our feature of a sweep. So when a sweep is ongoing, I've got lots of LD across a sweep. As the sweep comes close to completion, I get little LD around here. Why? Because I'm losing variation. I don't have much. So at a start, there's LD throughout a sweep site. At fixation, there's strong LD on either side, but not through a sweep site. And that gives rise to 2 different tests. One that gives you a signal for hard selection, one that gives you a signal for soft selection. So Kim and Nilsson's omega statistic basically does this. I take my putative site, I divide that chromosome into stuff on the left side and stuff on the right side. Then I compute the pairwise disequilibrium just among the elements on the left, the pairwise disequilibrium just among the elements on the right. Remember, disequilibrium is a correlation, so R is our estimated correlation. So I compute average LD among sites on the left side, average LD among sites on the right side, and divide that by average LD between a member on the left and a member on the right. That should give me a strong signal because basically I've got little cross-site LD, but lots of LD within a given site. That's a strong test for hard sweeps, but very little power for soft sweeps. Conversely, John Kelly's Z in S statistic, and S basically is number of sites that you look at, looks at average pairwise LD through a region. So I get a given region, I look at S segregating sites, and I simply compute all the pairwise correlations among those sites. This is a strong test for soft sweeps, but has little power for hard sweeps. So I have a site here. LD across that site is high for soft sweeps, but the partitioning of LD for hard sweeps is you have LD on one side, those sites are in LD, LD on the other side, little LD across those sites. That has to do with recombination happening early on as sites are going to fixation. They give different structures, and so here are 2 different tests based simply upon linkage disequilibrium. One works for hard sweeps, not for soft sweeps. One works for soft sweeps, not for hard sweeps. So you've got these LD-based tests. These are pretty standard, straightforward tests to apply. No questions about that? The LD structure is different in hard versus soft sweeps. And again, a soft sweep is also a partial sweep. So the partial sweep signal will be given by here. Because during a partial sweep, you're still from this region, so you have a lot of pairwise LD. As you move further along, the sweep gets close to completion, you get this trough, and you get LD on one side, LD on the other, but no LD across. The last category is a whole bunch of things, and these are tests based upon allelic age. And the basic idea, as I've mentioned several times, is under drift, a common allele is an old allele. So I can look at the age of the allele based upon its frequency and estimate its frequency under drift and contrast that with an independent estimate of its age. What about variation in STRs nearby? That allele appeared as one copy. If that allele is young, an STR nearby is going to have much variation. I can look at recombination between the site giving that allele and a nearby marker. I should have high levels of LD if the allele is young. That association will break up if the allele is old. So I talked about these earlier. I can estimate age by LD decay between a link marker and a site. I can look at the number of segregating sites around that haplotype, use that to age it. I can look at copy numbers at STR. I can look, for example, at the age of the mobile element. So the mobile element hops in, like the hopscotch element that gave the mutation giving TSNT branch 1. When many mobile elements hop in, they make a staggered cut in the DNA, stick the element in, so the DNA is bounded by staggered cuts. Those get filled in, so you've got exact flanking regions on either side generated by the insertion. You can look at the decay in those regions and get a signal. It's not very good because usually these repeat regions are tiny, usually, 5 to 10 nucleotides. But nonetheless, you can still get some sort of a weak signal from this. So I kind of talked about those methods before. I want to conclude by the broadest class of methods, which is long haplotypes. Alleles at moderate to high frequency with long haplotypes. Those can occur if you've got a region of low recombination, but you don't expect them to occur by most demographic features. A bottleneck isn't going to give you rare alleles at high frequencies. Is going to give you young alleles at high frequencies. Allelic surfing can, but other demography doesn't necessarily do that. So here's the basic idea. Here's time. I've got the purple denotes the SNP I'm using to define the allele. I can look at the haplotype around it is basically what's the conserved region, the LD block size about that allele. Over time, as that allele gets older, and therefore its frequency goes up under drift, the haplotype around it should get smaller. So common alleles under drift should have short haplotypes because there's been longer time for recombination to end. A common allele with a long haplotype is a good signal for selection and is rather robust to most features of demography if the geographic size isn't changing. Like a population bottleneck or expansion isn't going to give this feature, but a population whose range is expanding, you can get this feature if a mutation occurs on the edge of that expanding wave. It will give you a similar signal. So there's a bunch of tests in here. Only going to talk about a couple of them. So Sabetti was the one that sort of started this approach, looked at the ratio of haplotype lengths in two alternate alleles. So one way to control for recombination differences is to take a SNP and look at different alleles at that SNP and then contrast the lengths. If you have a region of low recombination, that's going to impact all the alleles, and so that's an internal control for that. There's a bunch of others you've seen here. So the linkage disequilibrium decay, NHS, IHS. We'll talk about singleton density scores. And there's other approaches where you contrast not within the same population, but contrast alleles between populations. So these cross-population studies take a population you think is a benchmark. So, for example, the founder, the source of the domesticated population, and then take a domesticated population and compare alleles in here with alleles in there. So artists just like to put in here. So as you imagine, she's pretty. If you're listed as Person of the Year, you're pretty well accomplished. So when the Ebola outbreak came out, Time magazine, the Person of the Year was scientists involved in Ebola, and Pardis was one of the people who did this. So Pardis is the sort of person, if you had a movie about a scientist, you'd say, you're overdoing it, right? Because she's smart. She's also the lead singer in her own band called The 1,000 Days. You can look them up on YouTube. They have quite a bit of following. And on top of all this, she's also really nice. So don't you hate people that are incredibly accomplished, but they're really nice so you can't sort of hate them? And so if you ever want to feel insecure about yourself, you can just look up what she's done. So her idea was the one that really got these long haplotype tests started. And the key idea is something called the extended haplotype homozygosity. So I've got a core SNP that defines a haplotype. Then what I do is I take all those alleles in the population that have that same core SNP. I have a set of haplotypes, and I simply expand out, and I'll see how far along I have to go before they start disagreeing. And we can use that measure as the extended haplotype homozygosity. And usually what people do is they wait, they extend out until there's a 5% or greater chance that two random haplotypes are identical in all markers. So here's the idea. Here is the core region for 2 different alleles. So as I sample across here, these are all those alleles. So for example, all those alleles at this point, there's about, let's say, a 80% chance, a 50% chance that they agree. And down here, I get the 5% chance that 2 random alleles agree. I call that distance there the extended haplotype homozygosity for the dark allele. This region down here is the extended haplotype homozygosity for the open allele. That's one metric you can use. Another metric is called the IHS, or Integrated Extended Haplotype Score. The idea here, instead of taking that distance, take the area under the curve. You're capturing more information. So there are tests based upon EHH and tests based upon the IHS, so the length or the area under the curve. And the basic idea is what you do is you take an allele, you compute its average extended homozygosity, then you look for all other alleles at that and use those as average. And the other alleles then allow you to control for differences in recombination length. So these tests are exquisitely powerful for catching ongoing selection. Once the allele frequency gets to be about 0.7 or more, the power goes way down. So these are great tests for capturing stuff that's undergoing selection. And here's proof of concept. So G6PD is an allele that's thought to be involved in malarial resistance, and this is another gene involved in malarial resistance. So what they basically did is they took random alleles, they computed this score, and they computed allele frequency, and what you see here is this is clearly an outlier, and that's clearly an outlier. So this is their idea behind sort of ground testing this method. Again, defining an allele, looking at the length of that allele, and then contrasting that with the lengths of other alleles at that same locus. And that's the extended haplotype score or the integrated haplotype score. And that's where some of these tests come from. So for example, there's the extended haplotype score, and then the IHS is when you do the test. Yeah, right down here. Integrated haplotype score, basically this same sort of idea. So people can take this notion, and there are 2 other ways people have looked at this. And again, the idea is how to use this information. And a simple way to use the information that seemed to work out well in the simulation is this NSL, number of segregating sites by length. So instead of looking at how long that region of extended homozygosity is, instead of looking at the area under that region of extended homozygosity, what they basically did was they looked at the average number for a given haplotype of consecutive segregating sites shared by 2 random members. So I'm a member of that allele class, you're a member, I line us up, I look how many segregating sites around it that we share. Then I take me and I take a second allele, look at how many we share. I take you and that second allele, look at how many we share, and compute an average value. So it's an interesting metric because you're not looking at length, you're simply looking at how far you go out in terms of number of segregating sites. And it turns out the simulations show it's significantly more robust to recombination and slightly more robust to non-equilibrium departures from the IHS test statistic. So it's actually a more simple statistic to compute. It turns out to have a little bit better features. Along similar lines, a very recent thing is something called the Singleton density score. And this is where you would have to do sequencing, right? All these things depend upon sequencing. And the idea is you basically take a target haplotype, and you basically then, on that given haplotype, count out how far you go until you find the nearest singleton on either side. So you look at me, got to go out 3 kb to find a singleton that I share that no one else shares. And we take that same allele in you, and you have to go out 1 kb to share a singleton. And singletons arise because you have mutations occurring on the tips of that coalescent. So if you've got a star-shaped coalescent, you'll have a lot of singletons. If you've got a deeper coalescent, you won't have as many singletons. And so it's a measure of singletons. What you can basically then do is take this measure and turn it into a time measurement and look at the average of the ancestral versus derived, and the singleton density score turns out to be very nice, and you've got power to detect very recent events with with fairly small amounts of selection. And so you have a time scale that's usually too short for these other haplotype-based methods. So for example, what they showed here was a sample size of 3,000 individuals and a arrival of 70%. They could detect ongoing events with a 2% selection advantage. So it's an exquisitely powerful method for a particular problem. I want to detect super recent selection, and it gives me lots of power to do that. As selection occurs over longer amounts of time, you have to do the other tests. And that's the drawback with all these different approaches. As you change your time scale, you basically change the test. That's where there's this huge myriad of number of tests. And again, the book, I tried to put them in summary tables so you can kind of see what they are together. So when someone uses a test, very often they'll have a paper they've just recently read that uses a test, so they'll use that test. Or they'll have some other relatively random criteria they use. It's good to know what all these tests are and what their strengths and weaknesses are. So just to summarize, we talked about the extended haplotype. We've talked about the integrated haplotype. We've talked about the number of segregating sites, Singleton density scores. Then there are other methods I didn't talk much about, which basically use the same approach of comparing alleles, but take an allele in one population and compare it to its length in a second population. That's looking for population-specific divergence. Okay. So summary, blah, blah, blah. Yeah, so basically, we'll talk about the Abba-Baba test in a second. That's fun. So basically, what haplotypes do, haplotype data, is it's the most powerful type of test to detect soft sweeps and to detect ongoing sweeps. As the sweeps get close to fixation, the power goes away because the class you're contrasting, one class gets really small and most of the power goes away. So we've seen a variety of different tests, all based upon polymorphic information. It depends upon how you gather over a time series, over a population, a single sample in one population, contrasting samples across two populations. It requires the time scale you're looking at, if you want to use allele frequency or site frequency, if you want to use folded or unfolded. So not surprisingly, there's this huge myriad of tests. And so the bottom line is always, which test should I use? And there's no answer to that, because everyone's asking different questions. If you think you've got soft sweeps or ongoing selection, you've kind of caught it in the middle, haplotype tests are best. If you think a sweep has just been completed, then something like the E test, the site frequency test, may be fairly powerful. If your data is evolve and resequence, so you've got a series of one or more populations you've seen diverge over time, you may want to look at allele frequency changes. What you simply need to do is to be aware that these tests are out there, be aware there's no magic test, be aware that many of these tests are basically asking the same thing, and so it can get very confusing because different groups will promote different tests, either because they're involved in them or because someone in their group read that paper and could explain to everyone else, and therefore they use that test a bit. So it's a big, complicated literature, but I try to kind of extract the basic ideas from that. So Chapter 9 discusses all the polymorphism tests, and the way it's structured is it starts out with an overview of the basic ideas, then it drills down and talks about each particular test in detail and gives you some of the references if you want to go look it up. So if you're going to use one of these tests, what I'd recommend is go back over the notes and look at the overview, and then if you want to use a specific test like a third generation test, then go to the chapter and drill down and look at the write-up on that specific test and what some of the strengths and weaknesses are. And again, never use one test on your data. Use a variety of tests, and don't use a variety of tests to find out which one gives you the signal. Use a variety of tests to tell you something about your data. Your data took you a while to collect. Don't, spend 5 minutes doing it. Now, the advantage of marker-based tests is there's no information about traits. They're also time machines. I can go back in time and look at selection that may not be ongoing now. So that's a real advantage of these tests. But the disadvantage is you don't know what trait they're involved. Just because you get a strong signal, it's often hard to track down what the phenotypic effect that signal generates is. Because again, you can't simply look at nearby genes. It could be a regulatory element that affects something on an entirely different chromosome. So you entirely miss what the signal is. So these marker-based tests are nice because you can use a polymorphic sample with no biology, no traits, no measurements of fitness, and make inferences about past selection. They'll miss a lot of current selection. They'll miss most of past selection. They'll even miss ongoing selection if it's sufficiently weak or you're using the wrong test. But that being said, these tests have a lot of benefits as well. So you should be aware of the pluses and minuses. It's like on a blind date, right? You don't want to look at, what they post on their page. You want to look at a full summary of everything. I'm giving you the full summary of all the person's strengths and weaknesses so you can decide if you want to have that second date with them or not. So questions then about polymorphism-based tests. You want to see Abba Baba, huh? As you can tell. So I have a short lecture that what I'll do is I'll work inside out because that will now catch us up. The lecture basically is, what do we know about when we apply these methods? Let me give you the punchlines up front. Punchlines up front is when people apply these methods for human populations, they got very non-overlapping sets. Let's say goodbye to Abba and Baba, and we'll come back to various people here. So let's go back, bye-bye Abba Baba. We'll come to you a bit later on. So here's an early example that came out. So basically there were 3 studies, one based upon linkage to Singleton decay, integrated haplotype score. These are similar tests, and one based upon outliers into GMSD. And what this shows is the number of genes declared to be under selection in humans, 1,800, 500, 200. This shows the overlap. Of these 2, only 125 are overlapped. Of these 2, only 47 were overlapped. So very low overlap between sets, very low repeatability. Some obvious reasons. Number one, the tests test for different things. The LD test, the first 2, tests for ongoing selection. They can catch signals that aren't picked up by TGMSD. So you don't expect them to match up because you're looking at different time scales. What about, though, these are pretty similar tests. How come they're quite different? Number one, statistical noise, false positives. Number 2, sampling different populations, right? So if I sample one set of populations, another population may have a different history of selection. So lack of repeatability across these tests really isn't, unexpected, but just be aware you have that out there. So what I want to do here is jump into domestication. And the Abba, how many people have heard of the Abba Baba test? It's how you distinguish a Swedish rock group from a Japanese rustler, right? And the idea is in domestication, figuring out the origins of domestication is really interesting because we can have introgression of a domesticated line could get introgressed naturalized genes. And the Abba Baba test came up because basically Neanderthal genes were found in that first migration coming out of Africa. So Europeans and North Asians have them, but South Asians and Africans don't have them. So let's talk about domestication. When we talk about domestication, we want to distinguish between domestication genes. Those are genes involved in going from the progenitor to the original line. Those variants then should be found over all land races. Improvement genes may be more land race or group specific. Darwin made an important observation that domestication could have been due to conscious selection. I want to have larger ears in my corn, or it could be entirely unconscious and entirely a byproduct of human-induced change in the environment. And a good example of this is suppose that as early humans were eating and had giant scrap piles, suppose that wolves, which were less timid at the scrap piles, got more food. That's not humans trying to domesticate them, but the net result is you'd end up getting wolves which are more timid and more comfortable around humans just because those wolves would get larger food supply. So domestication could either be direct, intentional action by humans or changing the environment and therefore creating a shift, the organisms shifting. One big issue, too, is were there single or multiple origins for a lot of things? For some things, there are really good single origins. My favorite story is rice. Rice is the most political organism on the planet outside of humans. Why? China, Japan, India are rightly so extremely proud of rice varieties, and therefore it's really important to them whether indica or japonica was the original progenitor. So if you read these papers, you get strong evidence that it was indica, strong evidence it was japonica, and evidence that you had gene flow between the bunch of them. So how do you determine gene flow? Well, the Abba-Baba test does that. Don't you love their, silken bathrobes there? I've got the common bathrobe picture here. The Abba-Baba test is basically this. It's called Abba-Baba because what I do is I have an outgroup, I've got two sister species and a relative, and what I do is I look for an allele that in the outgroup is different than in the sister group. So the allele arose there. Then I'm looking at that same allele which occurs differently fixed in A and B. So in this junction here, I was segregating A and B. So one passion is I could see Abba, A, B, B, A, or Baba, B, A, B, A. These should be equally frequent if there's no integration, because it's simply random partitioning of that sequence. If there's a directionality, that indicates that I have gene flow from 3 into 1 or into 2, and in this case, I think it was Africans, Europeans, and Neanderthal, and it could basically show that there was an excess here of the Abba, so the Neanderthal and the Europeans sharing it, and a deficiency of the Africans and Neanderthal sharing it. So your test statistic is really simple. It's this. Look at the number of Abba cases, subtract the number of Baba cases, divide by the total. Extremely unfortunately, the authors called this the D test. In the same paper, they talked extensively about Tajima's D. Drives you crazy. Plus, you got a great name like Abba Baba, right? Why mess with that and call it a D test? So the D test, whenever you say the D test, be proud and call it Abba Baba, right? So the Abba Baba test is a test for integration. A really cool example of this, recently from Jim Mallett's group, is in the tropics you've got these heliconius butterflies, longwings. And longwings have these elaborate mimicry complexes where if you're a collector, it's really hard to tell species apart because they mimic each other, and yet if you look at Melpomene, for example, in Costa Rica, and look at Melpomene in Brazil, they look quite different. It's the same species. And what they showed when they sequenced it is these different competing species were swapping out genes for color patterns. Pretty cool, huh? And so these Abba-Baba tests, I think, have become more popular, basically showing signatures of gene swappage between lineages. Let me conclude with a couple of fun examples. When you think about domestication, you typically think about animals or plants. And by animals, things you can eat or big things. But the silkworm has been domesticated for a while too. So here's Bombyx mori, the domesticated silkworm. Here's the Bombyx it came from. See, there's a bit of divergence. So recently people have sequenced both these genomes, and it's a bit of an odd thing because they focused on coding regions. Scans based upon coding regions are, in my opinion, bad because I think most divergence between populations occurs at least as much at the regulatory, that is, non-coding region, as the coding region. But nonetheless, they looked at coding regions, and what they basically did was they looked at reduction diversity in Mori versus his ancestor, and then looked also where you had low that is negative to G minus D score, and they found 1,000 regions of interest covering about 3% of the gene. And from this, they suggested about 350 protein coding genes as candidates for domestication genes. 160 showed differential expression, and 90 were expressed in the silk gland, midgut, or testes. So remember, when you have domestication, you can have conscious selection for increased silk production. But you also have unconscious selection to being grown in laboratory conditions, crowding, handling, etc. So you get evolution not only of target genes, but evolution of genes that occur as you're modifying the environment as well. And maybe the last thing to leave you with is we think of domestication as basically entirely human, but it's certainly not. Imagine the cool things you could do looking at domestication genes for ants which grow fungal colonies for food. There's obviously interesting genetics involved in the fungi adapting to the ants, and likewise the ants using the fungi. So I think domestication is a really interesting area that we've only started touching on now, because a lot of domestication is sort of commensalism, and a lot of, I think, really interesting problems that are out there that go outside of humans and crops and domesticated animals. So I think that's a good place to conclude. This is our overview of studies based upon polymorphic data. When we get back after lunch, we'll talk about tests based upon divergence between sequences. Now, there are a small number of tests, but the cool thing is it opens up a much larger question, and the much larger question is, what fraction of fixed changes are adaptive? And we have some tools for trying to estimate those, and we'll talk about those in some of their limitations.